Morning everybody, and welcome to your lecture on molecules to metabolism. So in this unit we'll be exploring the idea of how different biological molecules determine the different types of chemical reactions that happen in our body. And as a result of that, we have a sum of all these reactions, which is what we term as metabolism. Now in the background of this slide you can see a very, very complex image with lots of chemical names on it. This is what we call the Sigma-Nicholson metabolic pathway model. And what it aims to show is all the different chemical reactions that are going on in your body right at this present time. Okay, so you can see it's very complicated. You don't need to know a huge amount about it, but simple enough to remember that metabolism is the sum of all the reactions that happen in a human's body, okay, or in an animal's body. So in this unit, we'll be exploring the ideas of looking more closely at lipids and what they're made of, looking at proteins and what they're made of, and looking, of course, at carbohydrates. So the structure of DNA was discovered in 1953, and since then molecular biology has transformed our understanding of living processes. The relationship between genes and the polypeptides they generate is at the heart of the science, and the central idea can be simplified to the idea that DNA makes RNA, and RNA codes for proteins. The information in the flow cannot be reversed, and the protein generated cannot change the RNA or the DNA. Often the approach of a molecular biologist is a reductionist one. What a molecular biologist does is they identify the steps in a metabolic pathway, break down each one into its component parts. This approach has been a very productive one, and our understanding of many processes like respiration and photosynthesis are good examples of the success of this approach. Organic molecules, especially proteins, are very complex and extremely varied, and hence organic compounds have a hugely varied role within and outside of cells. There is a lot about organic molecules and cells we still have not discovered or understood. However, some scientists think that the reductionist approach alone is ultimately limiting, and that molecules can have dual roles, e.g. melanin, which is a protein and is the pigment that colors both skin and eyes, and may also interact with each other in ways that a reductionist approach can overlook. Carbon atoms can form 4 covalent bonds, allowing a diversity of stable compounds to exist. Despite only being the 15th most abundant element on the planet, carbon forms the backbone of every single organic molecule. Carbon atoms contain 4 electrons in their outer shell to form 4 covalent bonds with potential 4 other different atoms, e.g. methane or CH4, which you can see here. Covalent bonds are the strongest types of bond that form between atoms, and stable molecules can be formed. Now, because of the stability of covalent bonds, large molecules with many bonds can be formed. E.g., titin is the largest known protein, and it contains 539,000 atoms with a chemical formula of C16,1,697,023. Well, you read it yourself. Okay, you can see in this image a very small portion of that. Now, the result of these properties is an almost infinite number of different possible molecules that involve carbon. Here are some more of the common types. Here you've got starch. We know that starch is a storage molecule. Glycogen, which is a storage molecule in animals, and starch is a storage molecule in plants. And cellulose, which is a carbohydrate used in cell walls of plant cells. Now, carbohydrates, a few key facts, only contain carbon, hydrogen, and oxygen. They are organic molecules, compounds consisting of one or more simple sugars. Monomers follow the basic formula of CH2O to the power of X. That means that there is always twice as much hydrogen as there is carbon and oxygen. So for instance, if you think about glucose as a monomer, okay, mono meaning one carbohydrate unit, okay, its formula is C6H12O6. Therefore, there's always twice as much hydrogen as there is carbon. Moving on to look at lipids. Here is lipids are a group of organic molecules that are insoluble in water but soluble in non-polar organic solvents. Common lipids include triglycerides. Okay, these can be fats, which are solid at room temperature, or oils, which are liquid at room temperature. They can also be phospholipids, such as the ones we find in cell membranes, and steroids, the type we find as chemical groups in the body. Now you can see here this is the formula of a triglyceride called stearic acid. You can see it's got a glycerol head, okay, consisting of carbon, hydrogen, oxygen, and then it has the 3 fatty acid tails, okay, consisting of a carboxyl group and CH2, okay. Monosaccharides, okay, sorry, moving back to carbohydrates briefly. Monosaccharides have a ring structure when they're dissolved in water. So if you put glucose into water, it tends to form this ring. Okay, now we've looked at pentose sugars before in DNA, and we can always label them, the carbon atoms within the molecule. So in the pentose sugar and ribose, we've got C1, C2, C3, C4, C5. And it's a pentose sugar because it's got 5 carbons. Now in glucose, which is a hexose sugar, meaning it's got 6 carbons, we still label them from the far right, C1, C2, C3, C4, C5, and C6. Okay. Okay, moving on to proteins and amino acids. Now we've already looked at these before. Proteins contain carbon, hydrogen, oxygen, and nitrogen. Now all proteins contain those 4, and additionally sulfur is a common component, but it's not present in all proteins, but we do find it in the R groups of some amino acids. Proteins are large organic compounds made of amino acids arranged into one or more linear chains. So again, remember we did a presentation on this. The uses of proteins include hormones like insulin, enzymes such as catalase, immunoglobulins, which are antibodies, and gas transport. So therefore hemoglobin would be an example of this. And you can see the different colors represent the different polypeptide chains in each of these. Now on the basic structure of this, we have the amino acids. And you can see the backbone NCC of alanine is the same as the backbone of NCC in arginine, and the same as the backbone of NCC in leucine. Now there are 21 different amino acids found in eukaryotes, and you can identify which of the structures are identical. You have the amine group, which is this part here, the NH2. Okay, we have the carboxyl group here, which is the COOH. Okay, and these are identical for all amino acids. You have a H coming off the central carbon, okay, and then we have the bits that are different, which we term the R groups. CH3 is what makes this amino acid alanine. This R group is what makes this one arginine, and this R group is what makes this group leucine. Okay, you can pause the video for a second. Now, which of these molecules represents ribose? And which is molecule B and what is molecule C? Pause it now and have a go. Okay, so we know that ribose is molecule D. Okay, it has the O at the top and then the 5 carbons. We know that molecule B, okay, looking very carefully, we can see the NCC. Okay, we can see this NCC backbone. We can see an amine group. And we can see a carboxyl group up here, and we can see a CH2 up here. So this is an amino acid. Okay, molecule B is a triglyceride. Okay, and A to me looks like glucose. Which structure here represents an amino acid? Pause the video and have a go. Okay, you should recognize the amino acid as D. Okay, the carboxyl group is the big giveaway here. Okay, you've got COOH here, and you've got NH2 on this side. Okay, again, pause the video, have a go at the question. Okay, so you should remember that this one is ribose. Okay, a generalized fatty acid. okay, is 1 here, and a generalized amino acid would be here, carboxylamine. Now, which two of these do you think are most similar in structure? Pause the video, have another go. Okay, well, you should make the link that probably these are the most similar in structure. These are both ring-shaped when in water. These are both monosaccharides. They're both carbohydrates. Okay, but one is a pentose sugar and one is a hexose sugar. Okay, now again, we've looked at condensation and hydrolysis reactions before. Nothing too new there. What we should be able to remember is that condensation reactions make bonds and release water. Okay, and the reactions that are involved in are what we call anabolic, which means they're building molecules. Okay, so protein synthesis is an anabolic reaction. Okay, so in that you can see that here's a molecule going to bond with another molecule, and you can see that this is the water molecule that will exist, and then they will both share this oxygen atom here. And that is an example of an anabolic pathway. Okay, now hydrolysis is the opposite. Hydrolysis breaks bonds and it splits water. Okay, so it splits water from the reaction. And this is known as a catabolic reaction. And those are catabolic reactions are ones which break down molecules. So digestion would be an example of this. So the breakdown of starch into glucose would be an example of a catabolic reaction. Now, all of these chemical reactions require enzymes, which are biological catalysts. Okay, so here would be an example of that. So this is a polysaccharide, and in a polysaccharide, water is going to be split, and water is going to go into this reaction. It's going to use water to generate monomers from this polymer. Okay, so here again is a little bit more of that. Okay, here's your anabolic reaction. Okay, putting together these two monomers. An enzyme, which is a synthase, which is going to, okay, water is going to be released, and they're going to join together as two to make a dimer. Okay, di meaning two. All right. The reverse of that would be water out and splitting these into two monomers. Okay. Okay, a couple of quick examples of this. Maltose synthase condenses two molecules of glucose into maltose, forming a glycosidic bond. Okay, here you can see glucose and glucose forming together through a condensation reaction. Here's the water molecule which is going to be generated, and they're going to share that bond there. And because it's a carbohydrate, the type of bond we call it is a glycosidic bond. Okay, or a 1,4 glycosidic bond, because it's carbon 1 from this molecule bonding with carbon 4 of this molecule. Okay, plus H2O at the end. So this molecule of H2O is released. Like here we've got two amino acids joining together. So the carboxylic group, the OH of this group, joining with the amine group of this group. Okay, forming a peptide bond. Again, water is being released or removed from the reaction. And then what's happening is we've got a bond forming between the carbon atom and the N atom of the next amino acid. Okay, so then the CO and the N join, and we've got one molecule. Now this is called a peptide bond. So remember, in carbohydrates, glycosidic, and carbohydrates, it's a peptide bond. And here's a very quick example of catabolism by hydrolysis. Here you've got a protease enzyme hydrolyzing a dipeptide into two amino acids, breaking the peptide bond. So there's two amino acids here, okay, and we put H2O into the reaction, and what we get and the enzyme, and what we get here is one amino acid and another amino acid, okay. And you see the OH from the water is joining here, and the H is joining the amine group on this one. Okay, so water is going into that reaction. Anyway, thank you for watching the lecture, and we'll see you later.